Hi everyone, in this lecture we will be talking about the process of converting DNA into protein. These will span two topics, namely transcription and translation. Let's first start by reviewing the central dogma. This process starts with DNA replication, in which DNA is copied and passed on to daughter cells. We have previously made a lecture on this topic, so please make sure to check that out. Next we have transcription. In the real world, when we transcribe something, we are converting the same language into another form. So you might convert spoken language and transcribe it into its written form. The same thing or a similar thing happens in the cell, in which one form of nucleic acid is converted into another form. In this case, DNA is converted into RNA. Lastly, we have translation. In the real world, when we translate something, we are converting one language into another language. And in the case of the cell, it converts nucleic acid or genetic information into proteins. So RNA is read and converted into proteins, or to simply put it, into amino acids. Before we proceed, let's first talk about genes and gene expression. Genes are sequences of DNA that can be converted into products, either protein products or RNA products. And in this figure, we can see that gene expression rates can be highly variable. Here we have two examples of two different genes. One gene is transcribed into many copies of RNA, and as a result, many copies of the protein are also translated. But the cell can also have a low expression rate of genes in which a small number of RNAs are transcribed from the original gene, and this results in a small amount of protein being produced. The next difference between RNA and DNA is in the sugar that makes up its nucleotide. In RNA, the sugar here is ribose, and in DNA, the sugar is called deoxyribose. That's why RNA is named ribonucleic acid for ribose, and DNA is deoxyribonucleic acid because of the deoxyribose. Finally, the last difference between RNA and DNA is in one of the nitrogenous bases. In RNA, we have uracil, and this is complementary to adenine. But in DNA, instead of having uracil, it has thymine, and this is complementary to adenine as well. So the base pairing in RNA is U and A, while in DNA, it's T and A. Now our RNA can adopt a variety of different forms, unlike your DNA, which can only adopt a double helix form. These different forms can be formed by two main mechanisms. First, we have conventional base pairing, in which a purine pairs with a pyrimidine. From our previous lecture, we learned that the purines are adenine and guanine, and in this case, for RNA, the pyrimidines are uracil and cytosine. So in this case, the conventional base pairings are adenine and uracil, and cytosine and guanine. But RNA can also adopt non-conventional base pairing, and these can happen between nitrogenous bases of the same class, adenine and guanine, and cytosine and uracil. By adopting a variety of forms, RNA can have catalytic properties that are not present in DNA. Let's now move on to RNA transcription. This process, just like DNA replication, occurs in the nucleus, and as a result of transcription, the cell creates an RNA transcript, which in all cells is single-stranded. Now, RNA transcription has a very similar process to DNA replication, in which one of the template strands is used as a template, and it also proceeds in a 5' to 3' direction, as we can see in this figure. At the same time, it also uses complementary base pairing, just like DNA replication. In this table at the bottom, we can see the different bases on the template DNA strand and the transcribed complementary RNA base. For adenine, the complementary base in RNA is uracil. Then for thymine, we have adenine. For cytosine, we have guanine. And for guanine, we have cytosine. The only difference in this case is that thymine is not existent in RNA. Next, let's talk about the main protein involved in RNA transcription. This process is mainly carried out by RNA polymerase. And unlike DNA polymerase, this protein is an all-in-one protein, in which first it can unwind the DNA, as we can see in this figure, and attach the building blocks of the new RNA transcript. In this case, we call those as ribonucleoside triphosphates. There are 4 types: ATP, which carry the adenine, CTP, which carry the cytosine, UTP, which carry the uracil, and GTP, which carry the guanine. Unlike DNA polymerase, RNA polymerase does not need a primer to start a new RNA transcript. But the caveat to this protein is that it cannot perform proofreading, and this means that the different RNA transcripts are very prone to having errors. In this figure, we can see that a new RNA strand can be synthesized before the first RNA has even been completed. Here we can see the DNA at the center, and there are different and multiple RNA transcripts that are branching out of this single piece of DNA. This leads to the creation of many RNA transcripts. This is especially useful if the cell would like to highly express a certain gene. Next, let's talk about the different types of RNA transcripts. We have 6 types. The main ones are first messenger RNA or mRNA, and these are the RNAs that code for proteins. Then we have ribosomal RNAs or rRNAs, and they form the core of the ribosome structure, and they can be used for catalytic purposes, in this case, for protein synthesis. Then we have microRNAs or miRNAs, which help regulate gene expression. Then we have transfer RNAs or tRNAs. They serve as adapters between mRNA and amino acids during protein synthesis. In other words, these RNAs carry the different amino acids and facilitate protein synthesis. Lastly, we have other non-coding RNAs, and they can have a variety of functions such as RNA splicing, gene regulation, telomere maintenance, and a few other processes. It's important to take note that only mRNA is actually translated into proteins. Moving on, let's talk about the different markers of transcription. These are specialized sequences in the DNA that tell RNA polymerase where to start and stop transcription. The first of these is called the transcription start site, and this is the beginning site of the gene. Next, we have the promoter region, which are the nucleotides directly upstream or ahead of the start site, as we can see in this figure. The promoter region acts as the attachment site for RNA polymerase. Now, in order to stop the transcription, the DNA also has a terminator or a stop site, and upon reaching this area, it causes the RNA polymerase to stop and release both the template DNA and the newly made RNA transcript. Before we move on any further, let's first talk about the different types of RNA polymerase. There are 3 types, but let's take note that only RNA polymerase II actually transcribes mRNA that is used for protein synthesis, and this is what we'll be talking about in today's lecture. The 3 types of RNA polymerase are called RNA polymerase I, which transcribes most rRNA genes. Then we have RNA polymerase II, and these are used for all protein coding genes, as well as microRNA genes and the genes for other non-coding RNAs, like those in the spliceosome, which we will talk about in a bit. And lastly, we have RNA polymerase III, which is used to transcribe all the other genes that we were not able to discuss. Next, let's talk about a very important group of proteins that are needed for transcription. These are called the general transcription factors, and they are accessory proteins needed by RNA polymerase II to begin transcription. The first one we will talk about, and most important one, is called TF2D, or transcription factor 2D. This protein is the first to bind to the promoter, and it acts as a landmark for the assembly of other transcription factor proteins. In this figure, we can see the start site and the promoter region just upstream of this start site. Here we can see the TF2D, and we can see that it binds to a specialized sequence which we call the Tata box. The Tata box is called this because it is composed of both T and A nucleotides, and we can find the Tata box approximately 30 base pairs before the start site. Once TF2D has bound to the promoter region, it then recruits another transcription factor called TF2B. Before moving on to the other general transcription factors, let's talk about the different transcription factor binding sites. In this table, we can see the different transcription factor binding sites for both TF2B and TF2D. For TF2D, the largest and most important transcription factor, there are actually 3 binding sites. One, which we call the Tata box, approximately 30 base pairs upstream of the start site. Then we have the actual start site and a region downstream of the start site. Next, we can also see that TF2B has a transcription factor binding site upstream of the Tata box, approximately 35 base pairs before the transcription start site. Here we can see the different DNA sequences for these different transcription factor binding sites. In cases where we see a slash, this indicates that either of these 2 nucleotides can be placed in that sequence. This table demonstrates that the sequences for binding sites may be found before and after the start site of our gene. Moving back to the general transcription factors, other factors also have to bind to TF2D in order to form the completed transcription initiation complex. These include TF2F, TF2E, and TF2H. Together with a bunch of other factors, they help recruit RNA polymerase II. Again, altogether, they are known as the transcription initiation complex. Once together, RNA polymerase is released onto the gene via the addition of phosphates by TF2H. Once this happens, most of the TFs, or transcription factors, dissociate and the transcription can begin. Looking in closer detail at the phosphorylation of RNA polymerase, we can see that this phosphorylation actually happens on the RNA polymerase tail, as we can see in this figure. Again, this launches the RNA polymerase and begins translation, but it also has another function, which is to allow the attachment of RNA processing proteins. There are 3 main proteins. First, we have the capping factors, we have the splicing factors, and lastly, the polyadenylation factors. The reason why these are located in this area is that once a new mRNA transcript has been created, these different factors are now in close proximity and can readily process this new RNA transcript. Speaking of RNA processing, let's talk more about these different events. All of these occur before the mRNA is released into the cytosol. In this figure, we can see the nucleolus, which is the transcription region in the nucleus. And we can see that there are pores in the nucleus where the mRNAs can exit. But before that, 3 things must happen to our mRNA. These are capping, splicing, and polyadenylation. And we will be talking about these one at a time. First, and these processes occur in all mRNA, we have capping, which is the addition of a methylated guanine or a 7-methylguanosine on the 5' end of your mRNA. We also call this as the 5' cap. Next, we have polyadenylation, in which the 3' end is trimmed and hundreds of adenine bases are added. Together, we call this as the poly-A tail. These 2 processes are necessary for the mRNA because they serve very important purposes. First and foremost, they increase the stability of our RNA. RNA in general is highly prone to damage. So our 2 structures, the 5' cap and the poly-A tail, help prevent damage to the ends of our mRNA. Next, they facilitate export into the cytoplasm. They can also mark RNA as mRNA, and they indicate that mRNA is complete and ready for translation. Without these two structures, mRNA cannot be translated. Before we proceed to the last type of RNA processing, let's first talk about the two important regions in our eukaryotic gene. These are introns and exons. Introns are long, non-coding intervening sequences that flank our exons, which are the actual coding sequences in our gene. As we can see in this figure, the exons are typically shorter than the introns. Now, when we have mRNA that still contains both introns and exons, we call that as pre-mRNA or precursor mRNA. Again, this contains both introns and exons. Moving on to the last type of RNA processing, we call it as RNA splicing. This is the process of removal of introns from our mRNA, and this is done by detecting specific recognition sequences in the intron. The whole process is done by our spliceosome, which is actually a small nuclear ribonucleoprotein, or snRNPs, or SNRPs for short. These are composed of both proteins and small nuclear RNAs, or snRNAs, that can have catalytic activity. This figure demonstrates how RNA splicing occurs, and we can see that the intron is cut in what we call a lariat formation. RNA splicing and even the presence of introns and exons are unique to eukaryotic cells, and this allows eukaryotes to have multiple RNAs be produced from a single gene. We call this as alternative splicing. Depending on the needs of the cell, the different exons can be arranged in order to form different protein products, and we can see that here in this figure. Finally, let's talk about the different requirements for cytoplasmic export of mRNA. mRNA must contain three things to be exported into the cytoplasm, and these are the poly-A binding proteins, which bind to our poly-A tail, the cap binding complex or cap binding protein, which binds to the 5' cap, and our exon junction complexes, which indicate that the mRNA has gone through the appropriate splicing. Once out into the cytoplasm, this mRNA can now undergo translation. mRNA is actually degraded shortly after release into the cytoplasm. This is because RNA has a very short lifespan. Let's now move on to the last part of our central dogma, which is translation. Once mRNA has been successfully exported into the cytoplasm, it is then read in groups of 3 nucleotides called codons. These codons are then translated into amino acids using the genetic code. The genetic code can be seen here at the bottom of the slide. There are 64 codons, but there are only 20 amino acids that are used by our cells, and this means that some of the codons are redundant. This redundancy ensures that multiple codons can actually code for a specific amino acid. For example, in this figure, we can see that the amino acid alanine, abbreviated by A, can be encoded for by 3 codons: GCA, GCC, GCG, and GCU. We can observe that all of these codons are similar in the first 2 positions. Next, let's talk about the transfer RNAs or tRNAs. These are specialized RNAs that carry amino acids and detect the codons in our mRNA. In this figure, we can see the structure of our tRNAs. On one end, the 3' end, we can see that this carries the amino acid. And in the center of this tRNA, we can see the anticodon, which is a set of 3 nucleotides that are complementary to the codons. And this is responsible for the genetic code that we saw in the previous slide. One thing we can observe in these anticodons is that the 3rd position is what we call the mismatch or wobble position, and this ensures that there is redundancy in our genetic code. In total, there are 31 kinds of tRNA molecules. Now, our tRNAs are sort of like taxis that carry their amino acid passengers. Now, once the amino acid has been used up, to create a new protein, the empty tRNA has to be reloaded with another amino acid, and this happens in the amino acyl tRNA synthetase, which is used to attach the amino acid to its corresponding tRNA. In total, there are 20 amino acyl tRNA synthetases because there are 20 amino acids being used by the cell. In this figure, we can see the process of how tRNA is loaded with a new amino acid. Next, let's talk about the most important structure that is involved in translation, and these are our ribosomes. Their main job is to bring together the mRNA and tRNA that contains the amino acid, and this structure is used to form the different amino acid chains, or our proteins. It is composed of first the ribosomal RNA, or rRNA, and these are used to catalyze the different reactions to elongate our proteins. It also contains a variety of ribosomal proteins that are used to stabilize the RNA. In this figure, we can see the different ribosomal proteins and rRNA molecules that make up our ribosome. Another thing we can observe in this figure is that the ribosome is actually composed of two units. We have the small subunit, which we can see here, and this subunit has the job of matching the tRNA to the codons in the mRNA. Then we have the large subunit, whose job it is to have peptidyl transferase activity. This means that it can catalyze the peptide bonds that are used to lengthen the amino acid chain. Taking a closer look at the molecular structure of a ribosome, we can see that it contains 3 binding sites. These are known as the A site, or the amino acyl tRNA site. This site is where the tRNA enters. Then we have the P site, or the peptidyl tRNA site, in the middle of the ribosome, and this holds the polypeptide chain. Lastly, we have the E site at the end, or at the left side of our ribosome, and this stands for the exit site, where tRNA exits. This figure shows us the process of translation on the ribosome. The first thing that occurs is that a tRNA that contains or is holding on to an amino acid binds to the A site. In order to remain there, it must be complementary to the codon found on our mRNA. If it is not, it is ejected from the ribosome and another tRNA takes its place until it matches the codon on the mRNA. Once a match has been made, the growing polypeptide chain is then transferred onto the new amino acid by the large ribosomal subunit, which we can see here in step 2. This elongates the amino acid chain. Next, the large ribosomal subunit translocates or moves one step forward. This transfers the already used up tRNA into the E site and the newly created peptide bond along with its tRNA into the P site. And it also leaves the A site empty. After this, the small ribosomal subunit catches up and also moves one step forward to recombine with the large ribosomal subunit. This ejects the tRNA and leaves the A site empty. Now a newly charged tRNA can bind to this A site, and this process keeps on going until the protein is completed. Now when it comes to translation, it is very important to set the reading frame of our translation. Starting at the correct nucleotide is essential to create the correct protein. The ribosome has to make sure that it starts the translation at the correct position in order to have a functional protein. In this figure, we can see that we have the same mRNA sequence, but we can read it at different starting points. And as a result, we can see that the amino acid chain that is created as a result is different between each reading frame. Now, in order to start at the correct reading frame, the ribosome has to find the start codon, and this is the codon AUG, which carries the amino acid methionine. Let's now talk about how translation is initiated and terminated. The first thing that forms in the cell is called the initial translation complex, and this is composed of the small ribosomal subunit, a variety of translation initiation factors, and the initiator tRNA, which carries our methionine. It is important to note that this is the only tRNA that can bind to the small ribosomal subunit without the large ribosomal subunit. This initiator tRNA binds to the P site of our small ribosomal subunit. Together, this complex goes around the cytoplasm and binds to the 5' end of the mRNA. This is why it is very important for the 5' end to have its 5' cap. Without that cap, this complex cannot bind to it, and this means that the mRNA cannot be translated. Once bound, the small ribosomal subunit then goes along the mRNA, locating the start codon, which again is AUG. This signifies the starting amino acid, which is methionine, and it ensures that the reading frame of the translation is correct. Once it has found AUG, the different translation initiation factors then recruit our large ribosomal subunit, and it attaches to the small ribosomal subunit. As a result, the translation can then begin, and the different translation initiation factors dissociate from the ribosome. Let's now talk about how translation is terminated. The ribosome goes along the mRNA until it encounters a stop codon. There are 3 stop codons in total: UAA, UAG, and UGA. These 3 stop codons do not have a corresponding tRNA, and instead, release factor proteins attach to the mRNA when these codons are detected. These factors catalyze the addition of a water molecule instead of an amino acid in the P site. This releases the amino acid chain from the ribosome, and once this happens, the ribosome then dissociates from the mRNA. One thing that is very important to take note of, especially when it comes to eukaryotic protein translation, is that eukaryotic mRNA is monocistronic. This means that one mRNA codes for only one protein. This is in contrast to prokaryotes, which have polycistronic mRNA. This means that one mRNA in a bacteria can code for a variety of proteins, and usually these proteins belong on the same enzymatic pathway. This figure shows us that multiple ribosomes can actually translate mRNA at the same time. We call this as a polyribosome or a polysome. So as a result, we can create many copies of the protein in a short amount of time. Alright, so that would end our lecture on transcription and translation. If you would like to learn more about the things that we discussed, please make sure to check out these 2 references. Thank you very much, and if you found this video informative, please consider subscribing to our channel.